Start record. Today we're going to cover the second nervous and sensory lecture. It's going to focus on things beyond the synapse, what happens after the chemical messengers act on another cell. I know many of you guys couldn't make it last night. Apologize for that kind of odd time. It was just the way it worked out. But I did post, hopefully, or I did send you the link. Hold on a second here. The link, the YouTube link for yesterday's lecture. Hopefully you got that, had a chance to look through it. So today we're going to do nervous and sensory 2. Today's Thursday, so next week we'll be starting muscle, doing some of the anatomy of the muscle, physiology of the muscle, some of the changes that can occur with exercise, and some very interesting modifications in how muscles work. There is going to be and I will be posting those lectures sometime over the weekend. Next week is also our exam, which is, according to the syllabus, is on June 4th, which is Thursday. I am going to be sending you out a study guide over the weekend, and we will actually go over the study guide on Wednesday. So by you receiving the study guide over the weekend, that'll give you a chance to go through it. And if there's any questions you might have upcoming, either during the exam review or other times, you can certainly ask me. All right. And let's see. And all right. So if there's no questions, and again, feel free to stop me if there are, we're going to jump right into the nervous system. And I'm assuming before we start that everyone's been able to get access to everything on Blackboard. If there is a problem getting access to the online textbook or whatever, again, please let me know. All right, so yesterday or last night, we were discussing the nervous system, at least the intro to it, talking about what the general function of the nervous system is, how it's organized between central and peripheral nervous systems. We spoke about the anatomy of the different cells, then the functions between neurons and glial cells. We spoke about how nerve cells, when they're resting or not being stimulated, have a particular electrical charge associated with them, just like most cells do. We then follow that up by saying once a cell is activated from its resting state, due to a stimulus, it generates an action potential, which is an electrical signal that travels down the length of the cell, down the axon, and then ends up, the electrical signal reaches the end of that neuron at what's called the axon terminal, or the presynaptic terminal, something like that. And of course, that's where we have neurotransmitters or chemical messengers that are going to essentially exocytose. The vesicles are going to fuse with the membrane, exocytose the neurotransmitter, and that's going to interact with the next cell to continue or have an effect on the response, whether it's a muscle cell or other cells. 99% of all synapses are chemical. That's really what our focus is. Just like I said, with a chemical messenger or neurotransmitter being released across the synaptic cleft, interacting with another cell. And that's what our focus is. But I would be kind of doing you a disservice if I didn't mention that there are a small number of what are called electrical synapses. And essentially what an electrical synapse is the continuation of the electrical signal down the length of the axon across the gap junction to the next cell. We see this in heart cells, some types of smooth muscle cells, we see this. But compared to the number of synaptic connections throughout the body, it's very small as far as the type of seeing this electrical synapse. Most of the synapses we see are chemical, where, like I mentioned before, we have a neurotransmitter that's stored in the vesicle. When an electrical signal, the action potential comes down, that vesicle fuses with the membrane, squirts out the neurotransmitter, and has an effect on the neighboring cell. So this is really what our focus is going to be on: the response by the postsynaptic cell, the different types of neurotransmitters, and a few other things. All right. So what needs to be present at a typical, or what is present at a typical chemical synapse? What are the necessary ingredients, okay, that make up a synapse? Well, first of all, of course, we have the presynaptic neuron. This is really the source of the neurotransmitter. We have synaptic vesicles, right? We have this space, a synaptic cleft, and just for comparison, there is no synaptic cleft in an electrical synapse. Sorry about that. You can see there's really no, well, there's no space per se, because the electrical signal goes from right through this gap junction. It never leaves out into the space, whereas here it leaves into the space and then goes across. We then have the postsynaptic neuron or postsynaptic cell. On that membrane of the postsynaptic cell, we have two things. We have receptors, and these receptors are proteins that recognize the neurotransmitter and generate a response to it. We also have some enzymes. You don't see them in this picture, and these enzymes are important, as we'll see, in helping to recycle the neurotransmitter. We also have some what are called reuptake molecules, molecules that help to facilitate the recycling of the neurotransmitter. And what we can say without a doubt is that these chemical synapses are unidirectional, right? The signal is going to go this way. It's not going to go that way. So it's one direction. Synapses, if we're looking at neuron to neuron synapses, like here, because there are neuron to muscle synapses, neuro to gland synapses. If we're looking at neuron to neuron synapses, the synapse occurs either on the dendrites or on the cell body. There are exceptions where there are synapses between axons, but those aren't as common, and we're not going to go over those. All right, so this really shows the entire picture. Here we've got the presynaptic neuron with the vesicles. Here is the synaptic cleft, postsynaptic membrane with the enzymes, the receptors, and then reuptake molecules. So everything is all there. The only thing that wasn't mentioned on the previous slide, and this is because we find this not just with synapses, but other areas, is there's always a calcium channel that's present. And I'll take a look to show you the significance of that ion in nerve transmission. All right, so let's take a look at the series of events that take place from when a presynaptic neuron is stimulated. You know, think of like you touch something hot or whatever the stimulus is, whether it's a signal from your stomach that you're hungry. Whatever it is, we have this presynaptic neuron that generates an action potential. If you take a look, is number one. That action potential spreads, right, from the dendrite soma, it actually goes down all the way through the axon. It's carried down the axon to the axon terminal. And this is a very important step. Once that action potential reaches the axon terminal, it triggers the opening of voltage-gated calcium channels. Remember last night we spoke about voltage-gated sodium channels, voltage-gated potassium channels that play a role in an action potential. These voltage-gated calcium channels play a very important role in the exocytosis of the neurotransmitter. Here's the calcium channel. You can see it's opened up. There's like a little gate. Calcium then triggers the translocation of the vesicle to the membrane, where the membrane then the vesicle then dumps out its contents into the synaptic cleft, and then that neurotransmitter binds to receptors, and then we'll get a response, which is something we'll look at today as well. Now, I want to go through in a little bit more detail. I mean, as you know, you guys have taken several years of classes. You know that biological responses or mechanisms aren't just that simple. You know, calcium triggers synaptic vesicles fusing. There's got to be some intervening events taking place. And what is involved is the role of a couple different key enzymes. Once calcium enters the cell, it activates a couple key enzymes. One's called calmodulin kinase 2, and you could just spell it like that, or protein kinase A. Any enzyme that's called a kinase, it functions by phosphorylating. One thing we won't have time to look at this semester, but I'm sure you've heard, is the role of phosphorylation in any cellular process. Any cell that gets activated, whether it's glucose uptake, whether it's neurotransmitter release, whatever, phosphorylation, switch on, switch off, dephosphorylation. These kinases add a phosphate group to a protein called a synapsin. And it's the synapsin which actually guides the synaptic vesicle to the membrane. So calcium influx activates these enzymes, which in turn phosphorylate the synapsins, and synapsins actually act as kind of like a shuttle or a guide that translocates the vesicle to the membrane, as you can see. And then it's exocytosed, the neurotransmitter is. All right, so to kind of look at the picture, this is kind of a neat little diagram of some photomicrographs of immunofluorescence, which is a nice biochemical technique for identifying proteins under the microscope. And what this is meant to illustrate is the location of synapsins. So let's first take a look at the steps involved. Action potential travels down the axon terminal. Calcium channels open, calcium comes in. Calcium activates these enzymes, as I mentioned before, calmodulin kinase 2 and pKa. Those enzymes phosphorylate the synapsins, which triggers the exocytosis of the neurotransmitter. And that's kind of where we're at right now. This diagram is meant to show where synapsins are located in a resting cell and what happens when the cell is activated. In other words, an action potential. And let's just take a look at the green one, right? I'm sorry, green or red is fine. These are just different types of synapsins. The green represents the location of synapsins in the cell while at rest. This is identifying the location of synapsins when the cell is stimulated. And you can see synapsins move all throughout the cell upon stimulation, and then they return back. So I'm not going to ask you any details about these pictures, but I just want to show you how this is kind of an illustration of how these synapsins, their location and function changes, whether the cell is resting or being stimulated. Okay, so we're on the other side of the membrane now. So the neurotransmitter, whatever the neurotransmitter is, and we'll talk about many of them, are exocytosed across the cleft, and they reach the postsynaptic membrane. Now, one of the things that's commonly believed is anytime you talk about a neurotransmitter having in its role, we always talk about that it's stimulating something. It's stimulating muscle contraction, or it's stimulating some sort of response. Not all neurotransmitters are stimulatory. Some are inhibitory, and it really depends on the nature of the neurotransmitter itself and the type of receptor, which is something that we'll be talking about shortly. If a neurotransmitter is excitatory, okay, now you know we spoke about action potentials as being depolarization, repolarization, hyperpolarization. Well, there are certain circumstances where one event predominates. And in the case of excitatory responses, which we call an EPSP, this is associated with a dominating depolarization where there's a tremendous influx of sodium relative to everything else. An example of a neurotransmitter that is primarily stimulatory and primarily just causes depolarization would be glutamate. And we'll talk about glutamate a little bit later. Very important role, it's believed, in memory. And you can see this positive deflection of the electrical potential, right? If we're looking at a IPSP, or a neurotransmitter that inhibits a cell, that causes a hyperpolarization. Potassium leaves the cell. Some cases, chloride can come in. Either way, the cell becomes negative, and we see this. A neurotransmitter that is considered to have a negative effect or inhibitory is GABA. This is an amino acid that's actually associated with inducing us to sleep, the sleep response. And we'll talk a little bit more about that as well. So just to let you know, neurotransmitters can either stimulate or inhibit. For most of the remainder of the lecture, we're going to essentially just look at some different neurotransmitters, the classifications of them, how they work, and then we're going to end up with talking about one sort of chemical response involving the olfactory system, which is a direct application to how the primary, very important sense in animals. All right. So there's many types of neurotransmitters, and you can see they're classified 6 different types. We have acetylcholine. We have what we call biogenic amines. We have amino acids. We have purines. We're not really going to talk about those today. We have some neuropeptides and what's classified as unique molecules. It's kind of an interesting way to describe them. So we're going to go through not all of these, but some of these neurotransmitters, take a look at what types of receptors they bind to. Remember, the receptors are on the postsynaptic membrane, and how it generates a response in that postsynaptic cell. Okay. So the process of a neurotransmitter being released from a presynaptic membrane, binding to a receptor, and triggering a response in that postsynaptic cell is called signal transduction. Some of you guys may be familiar with this before in other classes. Now, there's many types of receptors, and receptors are unique for each type of neurotransmitter. So, for example, there's acetylcholine receptors. There's dopamine receptors, right? Like that. But if we take all of the receptors in general of every single neurotransmitter, we can divide them up into 2 main classes. What we call ionotropic receptors and metabotropic receptors. Ionotropic receptors, just for simplicity, these are receptors that are actually an ion channel. They just have a little space for the neurotransmitter or ligand to bind to. I'll show you some pictures of that. So they're like a ligand-gated ion channel or a ligand-gated channel receptor. This is one receptor that acetylcholine uses. I don't care about the molecular structure. They utilize an ion channel. What I do want you to know is this part, what we call a second messenger. Ionotropic receptors do not have a second messenger. Now, we haven't even talked about a first messenger yet, right? So if you think of a messenger, you think about, say, someone who comes by to deliver a message, right? In our discussion here, the first messenger would be the neurotransmitter. So acetylcholine, norepinephrine, whatever, they would be the first messenger. The second messenger is a chemical that's activated inside that postsynaptic cell. And there's 2 main pathways. And I'm just going to abbreviate them here, but then you'll see the full name. Second messenger pathways involve one of two different enzymes. And this is only found in metabotropic receptors. There aren't in ionotropic. We call it, one enzyme is adenylate cyclase, and the other one is phospholipase C. Okay, so let's back up for a second. Ionotropic receptors are ion channels that, when they're activated, ions go through, just like sodium, potassium, calcium. Metabotropic receptors are a different class. They're also known as G-protein coupled receptors. And once they're activated, this activates a second messenger, which is going to be one of these two enzymes. I don't really care about the type of synaptic effect or molecular structure or anything. Just know those two main different classes. All right. So this is a nice picture of an ionotropic receptor, right? This just looks like any other ion channel. The only difference is we have this little area that's carved out on the extracellular side of the membrane, and that is a binding site for a neurotransmitter. So you notice when the neurotransmitter is not there, this channel is closed. When the neurotransmitter binds, the channel opens. It's very fast response. In contrast, metabotropic receptors take a little bit longer, and I don't mean minutes or hours. I mean, instead of maybe milliseconds, it might take seconds or a little bit slower. This is our metabotropic receptor, also known as a G-protein coupled receptor, because we have all these different molecules called G-proteins that are linked to it. We're not going to have time to go through all of these, but just know that this is a different type of receptor. This is the space, this is the binding site for a neurotransmitter. In this case, when the neurotransmitter binds to the receptor, it activates the G protein. It also activates an ion channel, but it's a different one. So it's more of an indirect effect. In addition to that indirect effect on ion channels, it also will activate either adenylate cyclase or phospholipase C, which I will show you shortly. Okay, so to kind of summarize where we've been and so far, this is showing the exocytosis of a neurotransmitter. Remember, I mentioned that we have this influx of calcium, triggers the exocytosis of the vesicle into the synaptic cleft. And what we just learned, of course, with chemical synapses is that neurotransmitter binds to a receptor. It can either bind to an ionotropic receptor, as you can see right here. In this case, this happens to trigger the influx of sodium. Or it can bind to a metabotropic, which is abbreviated as GPCR, which is G-protein coupled receptors. And that activates a couple other events, whether it's opening ion channels downstream or also enzymes. And I'm going to show you some more detailed pictures, so I think you guys have a better understanding of it. All right. Now, this is kind of a daunting picture, but I'm just basically mentioning to you a couple things. This on the left lists the neurotransmitter and talks about the receptor and the receptor type. All I'm interested in you knowing from this diagram is these are the neurotransmitters that we're going to be talking about. These four: acetylcholine, dopamine, norepinephrine, and serotonin. Important thing to remember is dopamine, norepinephrine, and serotonin all bind to metabotropic receptors. So a little bit slower. In contrast, acetylcholine has two distinct receptors, what's called an ionotropic, and it also can bind to a metabotropic. And we'll talk about those as well. I'm not really concerned with these enzymes right here. We are also going to talk about the GABA neurotransmitter, that it's an ionotropic, it binds to an ionotropic receptor. Okay. So let's start from the beginning. Let's start with the first neurotransmitter, and that's acetylcholine. Acetylcholine can be an excitatory or an inhibitory type receptor. Good example where it can stimulate skeletal muscle contraction. A good example where it inhibits, it slows down the heart, right? It just depends on the type of receptor. This is the most abundant neurotransmitter in the peripheral nervous system. And the receptors, I mentioned we have two types, right? We have ionotropic and metabotropic. The ionotropic receptors are also known as nicotinic, and I'll explain why more detail a little bit later, but for now, just know that the molecule nicotine, say for example, it's found in cigarettes as well as other things, also binds to this same receptor, so it's called nicotinic. It's what we call an agonist. There's another compound called muscarine that's found in nature that binds to the metabotropic receptor. It's called muscarinic. The term cholinergic receptors just means that they're receptors that acetylcholine binds to. Okay. So let's take a look at how acetylcholine is made, first of all. Acetylcholine is made by the combining choline, and as you can see right here, the conversion of acetyl-CoA into CoA by an enzyme called choline acetyltransferase. So remember that enzyme. Choline acetyltransferase is the enzyme that's responsible for the synthesis of acetylcholine. That's how it's made. Now, as I mentioned before, once a neurotransmitter is released, it binds to a receptor, does its thing, but then I mentioned something about recycling or reuptake, right? There's an enzyme that it's abbreviated here, acetylcholinesterase. And let me, hold on a second, let me just go back. Sorry about that. Let me just, so ACHE stands for acetylcholinesterase. Acetylcholine, there you go. Esterase, there you go. This is an enzyme that degrades or breaks down and ultimately leads to the recycling of acetylcholine. So we have CAT, choline acetyltransferase, and we have acetylcholinesterase. All right. Nicotinic receptors, and this is something that's present throughout the animal kingdom, just to let you know. It's not just humans, animals have this too. The nicotinic receptor is ionotropic, as you know. It's a receptor that opens up, an ion channel that opens up when the ligand binds to it. Ligand is just another name for a chemical that binds to a receptor. So neurotransmitters or hormones, when they bind to a receptor, they're known as a ligand. In the case of the nicotinic receptor, this leads to an influx of sodium. And you can see primarily a sodium influx. Very simple, very fast. So stimulating muscle contraction and what we call preganglionic neurons of the autonomic nervous system. Postganglionic neurons of the autonomic nervous system, as we'll soon see, utilize a receptor that's inhibitory, which explains why it affects the heart, slows it down. But anyway, let's just stay on course here. All right, so it gets its name from the fact that nicotine is an agonist for it. And what is an agonist? An agonist is another molecule that binds to the receptor that triggers the same response. So nicotine binds to this receptor, triggers an influx of sodium, just like acetylcholine. In contrast, curare, if any of you guys have ever taken a class from Mike Bono, he's always has one of his legendary visual aids or visual activities is he talks about curare and with poison darts and things like that. Curare is a compound that's found in plants, but it's poisonous in the sense that what it does, if a person receives or basically ingests or is hit with an arrow, like a lot of indigenous tribes used to use, that contains curare, this will prevent the muscles from contracting, and they'll relax, which can cause a situation, especially with the diaphragm, where it is unable to contract, and this can cause respiratory failure. Although it's interesting, clinically, curare is actually used in very small doses to relax the smooth muscle sometimes of the trachea to allow for intubation. So it's true with a lot of compounds that were previously considered toxic, dangerous. Depending on how much is used and the situation, they can be beneficial. Whereas an agonist, in this particular case, you can see, in this case, here's the key into the lock, like the neurotransmitter. An agonist would be like this hairpin that can open the lock, different substance, but still does the same thing. An antagonist binds to the receptor, but doesn't allow anything to happen. So it blocks the effects. There is a pathological condition that's associated with the a defect in the nicotinic receptor. Oops, do I still have it? Oops, I guess not. And that's known as myasthenia gravis. It's an autoimmune disease in which the body builds antibodies that destroy the nicotinic receptor. So this can cause muscle atrophy, which ultimately can be fatal. Skeletal muscle atrophy. All right, so this is just another view of the nicotinic receptor. Now, I mentioned before that it's primarily an influx of sodium. Some cases you can get a small amount of potassium. It just depends on it. Just know that it's a ionotropic receptor, and it can lead to primarily an influx of sodium, in some cases an efflux of potassium. This is a nice picture showing the relationship between the nicotinic receptors, and here is the neuromuscular junction, which is, here's the membrane of a skeletal muscle cell, right? These are all the nicotinic receptor, and you can picture, here's the calcium channels. When the action potential comes down, calcium floods in, these vesicles fuse with the membrane, and exocytose the neurotransmitter, which binds to the nicotinic receptor. Now let's take a look at it up close. Here is, and you can kind of follow the steps, calcium influx, right? Ultimately, again, with synapsins leading to the translocation of the vesicle, exocytosis of the neurotransmitter, neurotransmitter binds to the receptor. And you can see here, if you look at the arrows, the influx of sodium dominates. There's a small efflux of potassium, but it's primarily a sodium influx. This ultimately does what? This ultimately, at the end of this response, this neurotransmitter will be inactivated by acetylcholinesterase, which will ultimately lead to the reuptake of the choline and everything, so it can be recycled to form new neurotransmitter. All right, so this is a picture illustrating myasthenia gravis. These green structures represent the nicotinic receptors. This is in a normal situation where a nerve releases the neurotransmitter, stimulates the muscle. This is in the case of somebody having antibodies, which are these Y-shaped structures. that are targeting and destroying the nicotinic receptor, there's no opportunity for the muscle to be activated. And muscle, of course, is one of those tissues that if you don't use it, you lose it, right? So there's definitely muscle atrophy that occurs. All right. Still talking about acetylcholine. Now let's take a look at the muscarinic receptor. Now the muscarinic receptor, there's many subtypes. Just because of how much we got to cover in the class and over a short time, I'm not going to go over the different ones, but just suffice it to say there's many different types. And this is a receptor that's associated with the autonomic nervous system, specifically the parasympathetic pathway. Now, you remember when we were discussing the nervous system last night, I mentioned that the autonomic nervous system has two branches, sympathetic and parasympathetic. In the case of the sympathetic, that's primarily known as the fight or flight, right? Speeds up the heart, speeds up breathing, right? Mobilizes us. Parasympathetic tends to kind of slow things down, slows down the heart, slows down the breathing. Acetylcholine binds to muscarinic receptors, and it mediates this type of parasympathetic response. So muscarinic receptors are associated with the parasympathetic pathway. This is a G protein coupled receptor, a metabotropic receptor that can either utilize phospholipase C pathway, which is a stimulatory effect, or an adenylate cyclase pathway, which is inhibitory. Now, there's a lot of information, right? A lot of details. So to kind of crystallize it a bit, if you think of acetylcholine in the parasympathetic nervous system, okay, at the same time that it slows down the heart and slows down the breathing, it's also stimulating the gut for digestion. This part of the autonomic system is known as the rest and digest, right? When we're talking about stimulating the digestive tract, that's the phospholipase pathway. If we're looking at inhibiting the heart, slowing it down, that's the adenylate cyclase pathway. I don't want you to worry about all this stuff and here, these pictures. I've got some other diagrams that really are a bit more, little bit more clear and refined. The agonist for muscarinic receptor is muscarine. Remember, it mimics the effect of acetylcholine. The antagonist for the muscarinic receptor is atropine. Now, many of you guys, I'm sure some of you have had some clinical experience volunteering or working in some hospitals or clinics. Atropine is a commonly used drug. It's a parasympathetic inhibitor, obviously an antagonist, by blocking acetylcholine. But how it's used clinically, it's used in everything like in a ophthalmologist's office or optometrist's office to dilate your pupils. I don't think they do it as much anymore with dilating the pupils, but when you go in, they want to look at your eyes. They give you those drops. Those are basically atropine, and atropine triggers this change in the musculature of the eye that allows your pupil to open up. It's also used in some types of surgeries to prevent people from throwing up after recovering from anesthesia. All right, so here's our muscarinic receptor. Remember I mentioned acetylcholine binds to a G protein receptor, which can cause, and remember with G protein receptors, they can downstream cause ion channels to open. By the same token, they can also trigger a second messenger, which can be acetylcholine or adenylate cyclase or phospholipase C. All right. So what ends the signal for acetylcholine? Well, we already went over that, but I just wanted to add a couple things. Remember, acetylcholinesterase is the enzyme that recycles or breaks down and recycles acetylcholine to be reused. But interestingly, there are certain products out there, pesticides, as well as nerve gas. Well, God forbid, hopefully we'll never be exposed to that. But in wartime, nerve gas contains substances that actually block acetylcholinesterase. So what does that mean? It blocks the breakdown of acetylcholine, which means acetylcholine is constantly hitting the receptor, which is basically keeping that tissue stimulated, which is not very good, right? With homeostasis, we want a response, and then we want things to go back to zero, go back to kind of where it was before. If a muscle is constantly stimulated, that's not good either, especially if you're looking at smooth muscle contraction, maybe of the breathing. And ultimately, it can be fatal. So when I say pesticides, that could be everything from in farms or even flea products. However, and I don't have time to go through all of it, but the more recent products that are used, the things like Program, Advantage, I can't think of some of the other ones. These are products that you can take your pet can take a pill. These are different family of compounds that they're called insect growth regulators. And they specifically don't have any side effects because it's a specific target that affects the growth and development of the insect. So they're definitely much safer than the old way of like flea powders and flea sprays. So yeah, so that's what many pesticides have. That's one of their unfortunate significant side effects, as well as like nerve gases. Okay, jumping into the next category, biogenic amines. And I'm gonna pretty much lump all of these amines into kind of one receptor, even though they each have their own individual, but they essentially pretty much operate the same way. There are 4 classes of biogenic amines, and you can see them right here. Amine means that they're derived from an amino acid. Dopamine, norepinephrine, and epinephrine are all derived from the amino acid tyrosine. There's tyrosine up there. Serotonin is derived from the amino acid tryptophan. In every case, these neurotransmitters bind to G protein coupled receptors, right? It can either be phospholipase C or adenylate cyclase, and you'll see that coming up. Now, this is where we see some of the differences, say, with the muscarinic receptor. So remember, the muscarinic receptor or any sort of acetylcholine response involves the breakdown of acetylcholine by way of acetylcholinesterase. These amines are inactivated by 2 different types of enzymes, what are called MAO or COMT, which I'll write out for you on another page. Interestingly, these amines are also, from a clinical standpoint, targets of certain medications to treat depression or even anxiety. You may have heard of serotonin reuptake inhibitors. They also have serotonin norepinephrine reuptake inhibitors. We even have serotonin norepinephrine dopamine reuptake inhibitors. These are medications that block these enzymes from working, which allows the neurotransmitter to stay in the synaptic cleft longer. Now, in this case, unlike those acetylcholinesterase inhibitors, like with pesticides, which can have a deleterious effect, this actually, by keeping the neurotransmitters in the synaptic cleft longer, this allows those neurotransmitters to be more effective, because it's believed and been shown in certain types of depression and anxiety that there's a disruption in the levels of these 3 compounds. All right. This is kind of a generic biogenic amine synapse. It doesn't show serotonin, but it could just as easily do that. Here we have our presynaptic cell. Here we have our postsynaptic cell. Here we have our synaptic vesicles. Here we have calcium, which would be coming in. Pretty much the same as any chemical synapse. Postsynaptic membrane, we have receptors, right? So much like what we saw before, calcium comes in, triggers the exocytosis of these of the neurotransmitter by way of the vesicles. The neurotransmitter binds to the receptor, triggers a response, which we'll look at shortly. And then those same neurotransmitters are inactivated by either COMT or MAO. And let me just pause there for a second. Let me go ahead and spell that out for you. All right. So MAO stands for monoamine oxidase. And COMT, it's a long one, catechol O methyl transferase, COMT. There you go. All right, so those are the two different enzymes that can inactivate and aid with recycling of the biogenic amines. All right, now let's take a look at some of the effects of some of the different neurotransmitters. And I think many of you guys might be familiar with this from other classes, but we're going to not talk about all of them, but I want to focus on a few. One is dopamine. Dopamine, of course, is derived from tyrosine. It's a very important neurotransmitter in the brain, the central nervous system. Where in the brain is it located? It's located in two main areas, what we call the nigrostriatal system, which is this area, and also the mesolimbocortical region. The dopamine in the nigrostriatal region plays a very important role in sort of smoothing out skeletal muscle movement. This is the area that's affected during Parkinson's. So if you've ever seen someone with Parkinson's or are familiar with it, you know they have tremors, right? Or shaking. This is due to the fact that the absence of dopamine and the inability to exert its effect affects the smoothness of muscle motion. The second area where dopamine has an effect is the mesolimbocortical area, which is really the prefrontal cortex. This, of course, is one of the latest areas of the brain to develop. This is important in executive functioning, as well. It's the reward area of the brain. Reward meaning if you do something you enjoy, whether it's getting together with friends, eating, drinking an espresso mocha, something like that, you feel good. One of the reasons why you feel good is you're enjoying it, dopamine's produced, and it makes you want to keep doing that. It's a kind of a feel-good neurotransmitter. And when you do something and you're satisfied, dopamine's produced. Unfortunately, this is also the neurotransmitter that's targeted by certain drugs that, because of that, are very addictive. Things like cocaine, Ritalin, and Adderall, which is used for people who have ADHD, drugs like heroin, even nicotine. Now, these drugs certainly have other effects, but they do affect the dopamine pathway. And when someone, that's why it's so difficult to withdraw off this medicine, because when you withdraw off this medicine, dopamine levels drop, and people will sometimes get, do whatever they need to do to get that that feeling back. So that's why otherwise maybe kind people will resort to violent actions because they need to get that dopamine feel back. The way that drugs can affect dopamine is they can do a couple things. They can increase the release of dopamine. So in other words, the more synaptic vesicles releasing more dopamine. And at the same time, some drugs also will block the reuptake. In other words, both cases will lead to elevated levels of dopamine in the synapse, but different ways. One, you're adding more to it. The other one, you're just not removing as much. So it's quite interesting. All right. Let's jump to norepinephrine. Now, even though all these biogenic amines are all binding to G protein receptors, we sometimes call the receptors more specific, like dopaminergic or dopamine receptors are called dopaminergic, even though they're G protein. Norepinephrine and epinephrine bind to what are called adrenergic receptors. They contain two different subtypes, A and B. You know, it's kind of like with acetylcholine, right? They have nicotinic and muscarinic. And of course, these subtypes will determine the response. Clinically, you may have heard of what are called beta blockers for people who have high blood pressure. Basically, beta blockers block the effects. They're an antagonist, block the effects of norepinephrine. If you think of norepinephrine as a fight or flight neurotransmitter or a hormone, the heart's working harder, pumping faster and stronger. And if someone already has high blood pressure, you want to kind of dampen that a little bit. So that's why beta blockers are used. Lastly, let's talk about serotonin as far as the amines. It's derived from tryptophan. We still have the same, same ingredients, vesicles. We got calcium coming in. We've got MAO and COMT involved. Okay. I want to mention a couple of things, though, that are really significant. They have a wide range of actions affecting mood, behavior, appetite, and cerebral circulation. And what's interesting is certain medicines that have been used, known as like serotonin receptor agonists. One particularly is called sumatriptan or Imitrex. Those of you that get migraine headaches, you may be familiar with this drug. This is a drug that's used to level off or to reduce migraine headaches. Because serotonin, what serotonin causes, it causes a change in the dilation of blood vessels, even in the brain. And so people who have migraines, you can give them a drug that restore or changes that circulation so it doesn't put as much pressure. Different causes of migraines, but that's one. Most of you guys, not all of you, are a little too young to remember this drug. You may have heard of it, though. It was called LSD. It was kind of considered to be the psychedelic drug of the 60s. Interestingly, in some psychiatric circles, they're using this in small amounts for people who don't respond to conventional antidepressants. So there's drugs that stimulate serotonin, and of course, we also have SSRIs, which are reuptake inhibitors, which block the reuptake of serotonin, which keep serotonin levels elevated. All right. Now we're just going to kind of push through a little bit more quickly with some of the others. So we've gone from the class of neurotransmitters, acetylcholine, to biogenic amines, now amino acids. A couple I want to mention. One is glutamate. This is an excitatory neurotransmitter that appears to play a role in memory storage. Again, if you had more time, I'd talk to you about a professor I took at UC Irvine. His name is Gary Lynch, L-Y-N-C-H. I was really blessed to have him. He's made some really fascinating discoveries back in the 80s and 90s. After I left, actually, he was a good teacher in the 70s as well, but linking glutamate and other messengers to memory. If anyone's interested in finding any more about him, There's a great book about him. It's called 101 Theory Drive. 101 Theory Drive, that was the address of his laboratory. And it's a really interesting story about life in the lab. Really cool stuff. There may be a disruption of the glutamate pathway in people who have Alzheimer's. Not sure, a big question mark. GABA, another neurotransmitter, this is inhibitory, which means that the cell becomes negative as opposed to being depolarized, mostly due to an influx of chloride. This receptor is this is a, you can see this is actually a ligand-gated ion channel. This is a receptor that's targeted by certain types of tranquilizers, what are called benzodiazepines, that help people sleep. You may have heard of a drug called Lunesta. They used to actually have a lot of commercials on it late at night with that green moth or butterfly flying through the air and talking, and it can kind of make you sleepy just watching it. But this is a particular drug that binds to the GABA receptor and in many people help to induce sleep. So a lot of medications associated with binding to receptors. A couple others, like I said, I'm just going to kind of plow through very quickly. Neurotransmitter called CCK is important in giving us, lets our brain know that we're full, that we've had enough nutrients, that we're satisfied, we're satiated. Very important neurotransmitter, and certain pharmaceutical companies are working on agonists of this for people, for example, obviously with the concern with obesity, overeating. Substance P, easy to remember. P, think of pain. This is a neurotransmitter that when it binds to its receptors, triggers the pain sensation. Now, there are other ones that do it as well, but this is an important one. One interesting feature is, you're probably wondering why I'm showing this chili pepper. Well, chili peppers produce capsaicin, and the amount of capsaicin in that pepper correlates with how strong that pepper is. And there's a scale. I can't think of the scale offhand, the numerical scale, but the higher the number, the more capsaicin and the hotter it is. Capsaicin actually can deplete substance P. So you might wonder, well, this doesn't make sense. It's like when you put the pepper, if you put a whole pepper in your mouth is on fire. Well, if you notice people that be eating peppers a lot, say from childhood, they're able to eat peppers very easily. So they're desensitized. Capsaicin clinically is also used. If you go to the pharmacy and say, there's a couple different products, but one is called Capazol. There's these ointments, creams that people can use to spread on, say, sore areas like your back or other areas. Those ointments have capsaicin in it. And it's been found that maybe initially it might burn, but after a while it will reduce the pain sensation because it's depleting substance P. So anyway, interesting bit of trivia there. All right. Neuropeptides. So these are peptides, chains of amino acids that are found in the nervous system. A couple to mention. One referred to as the endogenous opioids. You've heard of opioids or opiates, right? You've heard of the concern with the opioid crisis we have in this country with fentanyl, things like that. Well, we produce our own morphine-like substances. One example is beta-endorphin. You've heard of endorphins. They also block the release of substance P. If you've ever noticed, like when someone has the runner's high or whatever, they don't feel anything. They're like almost like euphoric. Has an analgesic effect, reducing pain and kind of also giving you a sense of well-being. We actually have opioid receptors in our brain. ADH and oxytocin. These are a couple different hormones that are produced in the hypothalamus. And we'll talk about these more when we go to the endocrine system, but many of you have heard of oxytocin. This is a smooth muscle, stimulates smooth muscle contraction primarily in the uterus during childbirth. ADH is a hormone that promotes water uptake when we're in a dehydrated state. Finally, we have neuropeptide Y, which helps to stimulate appetite. So whereas CCK and other hormones as well, or the neurotransmitters, suppress appetite because we're full, we don't feel hungry, neuropeptide Y gives us the sense that we're hungry. So later on, hopefully if you have time this semester, I'll talk to you about kind of the endocrinology and the physiology of appetite. It's really quite remarkable, actually, how it all works. Lastly, the unique neurotransmitters. We produce what are called endocannabinoids. These are neurotransmitters that bind to what's known as the CB1 receptor, which is the same receptor as THC, which is the active ingredient in marijuana. I'm sure you're familiar in the news these days with cannabinoid oil and obviously medical marijuana and things like that. Well, the reason why there's medicinal value is because we have these receptors. It has an analgesic effect, also an appetite stimulant. And of course, there's a lot of different ingredients in the plant, and it depends, certain types are more hallucinogenic, certain ones are just medicinal. Excuse me. The last one looks like I'm saying no, but it stands for nitric oxide. This is actually a gas. It's a gas that's released from some neurons that triggers smooth muscle relaxation, vasodilation, right? So the reason why our blood vessels constrict and dilate, depending on our physiological state, has to do with the fact that they have smooth muscle. And there's very important in terms of regulating, for example, our blood pressure and things like that. This has actually been the target of certain medicines. Nitroglycerin, which is interesting. It's one of the active ingredients in explosives, but it's also a drug that was used. My grandfather used it. When nitroglycerin enters into the blood, it decomposes into nitric oxide, which causes the blood vessels to dilate. In his case, he was getting angina, which many of you know, it's due to kind of a ischemia or reduced blood flow of the coronary vessels. By taking nitroglycerin, it would dilate them and he would get better blood flow. More recently, there's a couple drugs such as Viagra, Levitra, Cialis. They do the same thing. However, they're more specifically targeted for a condition called erectile dysfunction, which this helps to increase blood flow to the arteries of the penis. Although it's also used in women as well for sexual response. All right. So I'm going to finish off here. Let's see how we're doing. All right, we're doing good, hopefully. All right, so now I'm going to kind of jump into, I wouldn't say jump into a new area. I'm going to branch out. And we've spoken a lot about receptors, right? For example, receptors in terms of that trigger a response of some sort. We spoke about G protein coupled receptors that respond to neurotransmitters. We've also spoken about or receptors that say if you touch something hot, right, this activates the nerve pathway, right? There's receptors there that they're going to respond to a stimulus. And you can see there's a whole battery of different receptors, and their names indicate what they respond to. For example, mechanoreceptors respond to kind of movement, touch, pressure. Osmoreceptors respond to osmolarity of the blood. Thermoreceptors respond to hot or cold. Taste receptors, chemoreceptors respond to, again, dissolved food material, sweet, bitter, different ones. We have olfactory receptors, which are receptors that are activated when what we call an odorant or a smell binds to the receptor. We have photoreceptors, of course, that are found in the eye and other areas. So what I want to focus on today is olfactory receptors because, again, just to show you kind of the relevance of this. All right. So when we talk about olfactory, first thing we think of is the nose, and this is true in all animals, right? We all have, for example, here's our nasal cavity, here's our brain. In the mucosa, or in the lining of the nasal cavity, we have what are known as olfactory receptor cells or olfactory epithelium. And when something is brought in, when you smell something, this will activate or trigger these receptors that will send a signal to the olfactory bulb in the brain, and it'll be interpreted. Is this a pleasant smell? Is this like a, nice juicy steak? Or is it something that's not very pleasant? So let's zoom in up close. All right. So where we're looking at is we're actually looking right in this area. So these are these nerve endings, the olfactory cells, and this is the olfactory bulb. If we go here, this big area is the olfactory bulb, right? These are those little like hairs, right, that we saw right there. Okay, so the olfactory pathway, we call this the vertebrate olfactory pathway because it's the same in us as it is in dogs, as it is in other animals, although some are better than others because the number and the types of receptors they have are better, but nonetheless the pathway is the same. And the pathway involves an odorant, which is the stimulant, which activates these receptor cells. These receptor cells then communicate with, you can see this olfactory bulb, this very long cell. So here's this olfactory cell, goes all the way up into the olfactory bulb and communicates with the olfactory bulb, and that signal then sends to the brain, rest of the brain where we interpret it. It's like, that smelled good. It's like, oh, that smelled horrible. All right. So let's take a close-up look at a olfactory receptor cell. And this is like a little experiment they did. They took an olfactory receptor cell, which is essentially just like it's just like a neuron, really, because if you think about, here's the axon, right? And this would be equivalent to like the dendrite cell body, just a very specialized one. They did an experiment where they took a pipette and squirted out a particular odorant, or a particular odorant is just another name for a molecule that generates a smell, whether it's a smell like from a rose, right? Or a smell from, I don't know, just other nasty smells or whatever, or pleasant smell. What happens, then they did some electrical measurements, and they found that when the odorant binds to the receptors on the receptor cell, it triggers these interesting electrical changes. And if you remember from last night, I showed you that this is what we call patch clamping. Every time you see this downward deflection, this indicates an opening of a channel. So in addition to what we just looked at, the whole time talking about neurotransmitters and receptors and ion channels and things like that, the opening of ion channels in response to a smell occurs as well. So it's very similar. Now let's take a close-up look as far as what type of signaling pathway is involved. The olfactory receptor is a adenylate cyclase or a metabotropic receptor. And we're not going to go through all the details, but what I do want you to know is that when an odorant binds to this olfactory receptor, which is a G protein receptor, we see the activation of this enzyme called adenylate cyclase. And adenylate cyclase will ultimately lead to the influx, as this you can say, of ions, some calcium, but let's just focus on sodium. So I just wanted to illustrate, giving you a specific example of a particular type of sense, in this case, smell, is governed by the same signaling mechanisms as we think of with typical nerves. In this case, it uses the adenylate cyclase pathway. Now, I want to expand a little bit and take a look at kind of an accessory olfactory organ, and this is present in a variety of animals. Here you can see a rodent. It's also present in sheep and goats, and I'm sure a wide range of other ones. In addition to that typical olfactory bulb that we saw in the brain, they have an accessory olfactory bulb. There's the main one, and these animals have an accessory bulb. And they've got a separate set of neurons that goes from the olfactory cavity, and actually even the oral cavity, that goes up to that olfactory bulb. In this case, they utilize both, in this case, they use the phospholipase C pathway. And why is that doing that? This is interesting. Okay. I don't know why it's doing that. Oh, I know. I was wondering why it's showing it twice. Oops, let me back up for a second. So, whereas with the typical olfactory receptor, picks up signals, generates that electrical impulse via the adenylate cyclase pathway, communicates with the olfactory bulb. With the vomeronasal organ, we have nerves that are found in the nose, but also in the oral cavity that send signals to this other accessory organ, and the communication pathway is the phospholipase C. This V1 and V2 just indicates that there are two different types of receptors with vomeronasal organ, but you don't have to worry about those. And it looks like they both use the same pathway anyway. All right, and this is just a close-up view of that. And this is just showing it kind of close up. All right, so let me go ahead and, oops, what happened there? Let's see, okay. All right, oh, we got 13 people here. Okay, so that takes us to the end of today's lecture. We're going to start with muscle next week. Like I said, I'll be sending you out the updated muscle lectures over the next couple of days along with the study guide. At this time, does anybody have any questions? And you can say it out loud, or if you want to type it, that's fine too. Okay, well, thanks for coming and have a good weekend, and I'll see you next week.